I really just want to welcome you all to Translation 2025 and the ECR morning. It's really lovely to see you. Most of you know who I am. I'm an associate professor at the University of Oxford, the Department of Psychiatry. I'm also senior scientist for Dementia's Platform UK. I've been working with the director, John Gallagher, for the past nearly 9 years now, and I lead various scientific programs using resources from Dementia's Platform UK, from the data portal. And it's really an honor to again introduce sessions for ECRs, and I really do enjoy this part of my role here. So really welcome, and especially those who've traveled far today. I know we have colleagues from South Korea, so a welcome. It's really lovely to have you with us today. And thank you also for all of you for your poster submissions and lovely posters, and we really do appreciate the time you've taken to put together those posters to reflect your scientific work. So thank you all very much, and I really hope you'll enjoy this morning and the conference. So I'm going to speak very quickly today, and then I'm actually going to hand over to a few of my researchers who are going to do some introductions today. This will help you not get bored of my voice, but I also get bored of my voice. So I will introduce a little bit and they will take over from me. Basically, the program today, we are very, very honored to welcome two of our sponsors and partners from Alzheimer's Research UK and also the Alzheimer's Society, that is Jacqui, Hanley and Cherie, Nyota, who will be talking about opportunities at both these societies for funding and research. We are always very, very grateful when they present at our conferences, and I'm sure you will really enjoy what they have to say and take away valuable information. We will also have a data portal overview by Emma Squires from Swansea University, and I will cover some training and mentoring opportunities that we are offering through DPUK. We will have 2 ECR lightning presentations, and we will also have 2 career presentations by senior researchers who've taken very different career routes, but show you the different options that are available for researchers in science. And we are very honored to welcome an address by Professor John O'Brien from the University of Cambridge. And then we will also have opportunity for a Q&A at the end where we can ask the panel. So rather than have questions throughout, we will have a panel at the end. This will enable us to have more questions. So please do store up your questions for the end, and we will make sure, hopefully, that your questions are answered. So as most of you are aware, DPUK is not just a data portal, but it is an organization that facilitates dementia research through providing access to secondary data sets, whether that is imaging, genomics, or clinical or population cohorts. We also enable training for researchers. And as I mentioned before, I run my scientific programs at Oxford for the last nearly 9 years, entirely on the data portal. I've been awarded grants, fellowships, and these programs of work use the resources of DPUK. So I have 2 hats, my DPUK hat, but also my scientific hat. And if we have a look here at this diagram, we can see that the data portal provides rapid data access, multimodal data with a flexible configuration, data curation and training. Emma will talk to this much more widely during her talk. We also have a trials delivery framework. You'll hear more about this during the conference. And we have an experimental medicine incubator. So these are what really makes up DPUK. And we really are here to serve the research community as a whole. and most importantly are our industry and academic and philanthropic, wow, that's a difficult word, partners, without whom we could not exist. Although our core funding is by the Medical Research Council, without our partners, we simply could not do what we've done for the last 10 years and hopefully going forward as we go into renewal. So that's enough of me. I would like to welcome our first speaker, who is Jacqui Hanley from Alzheimer's Research UK, who is going to talk about opportunities available at ARUK.
Hi everyone, as Sarah said, my name's Jacqui. I'm Head of Research Funding at Alzheimer's Research UK, and I keep saying I'm relatively new. I've probably been there nearly a year now, but one of the things that attracted me to work at Alzheimer's Research UK was the work they do around early career researchers and supporting ECRs in their career. So I'm really pleased to be here today. So I'm going to talk a bit about kind of what we do, who we are, what support and opportunities we offer, and how you can work with us. So, there we go. So for those of you that don't know us, Alzheimer's Research UK is the UK's leading dementia research charity. We're all in the dementia field, so we know that 1 in 2 people will be affected by dementia in their lifetime, either by developing the condition or caring for someone with dementia. Our organisation exists for a cure. That's our mission, and we see our vision is a world free of the fear, harm and heartbreak of dementia. So we kind of do this across 3 main areas, 3 goals in our strategy. So we've got treatment, diagnosis and prevention. So treatment is making sure that people get access to new life-changing treatments for the diseases that cause dementia. Diagnosis is all about people getting earlier and a more accurate diagnosis for improved access to treatments and to research. And then prevention, it's making sure that fewer people develop the diseases that cause dementia. And there's a few ways that we work across these 3 areas, but it's also important to acknowledge that you sometimes might see a 4th area, 4th pillar, because it's really important to acknowledge that progress against these 3 goals is driven by progress in the fundamental understanding of the biology of the diseases that cause dementia. So you might sometimes see a 4th pillar of understanding, especially if you apply to us for funding. There's a tick box for understanding. But as I said, we work across these 3 main areas in lots of different ways. Diagnosis, for example, with the Alzheimer's Society and People's Postcode Lottery, we fund the blood biomarker projects. And I know we'll hear about Readout that uses the DPUK network throughout the conference. But yeah, that's kind of who we are, what is our strategy, how we work. But I know what you're most interested in is kind of how we can support ECRs and funding that we offer. It's just important to note that we fund kind of fundamental, basic biology, biomedical, clinical research, obviously across those 3 or 4 goals. Social and kind of carer-focused research is not within our remit, so that's kind of more for our colleagues at the Alzheimer's Society. But we fund lots of grants across the ECR pipeline. So we've got PhD studentships or scholarships or the clinical equivalent through to kind of research fellowships, senior research fellowships, and then kind of project grants and major grants. We also fund bridge funds. So these are kind of small pots of money up to 30K for 6 months full-time or longer part-time to kind of help bridge those gaps or those pinch points that you see when you're kind of moving through your career. Lots of different types of grants that we offer. And I'm not going to, I do have slides that kind of talk about each and every one and kind of the amounts you can apply for, eligibility, that sort of thing. But there's way more information on our website and there's a lot more detail in there in terms of kind of what you can and can't apply for, who needs to kind of be part of the grant. What else is there? That's kind of it, really. One thing to note on our fellowships is that there's no time limit in terms of when you can apply from when you've had your PhD. So there's it's only our Race Against Dementia fellowship that we fund in partnership with RAD, that you have to apply within 6 years of getting your PhD. But all of our other fellowships are kind of not time bound, which I think is really important. We've just closed our rounds. It was literally last week that we closed our fellowship round, so we've slightly mistimed this. But we will be opening our clinical fellowships in the next couple of months. And do look out on our website for kind of future rounds. The bridge funds will be open soon. And so will some of the other projects. I recommend signing up to our newsletter. That kind of helps to keep track of when things are open and closing, and other kind of opportunities and support that we might offer. And then in clinical research, there's our two clinical fellowships at the moment that are kind of for a PhD and post PhD. But again, way more detail and information on our website. One thing to flag is we've got an early career researcher portal. It's in the process of being updated and kind of rebranded, but there's lots of information on there in terms of, the funding opportunities, resources, tips about how to talk about your research, how to write a lay summary, infographics and sort of dementia statistics, kind of how to, as I said, kind of how to talk about your research and engage with kind of the community and patients and the public. So please do check it out. There's lots of really useful info on there. And just in terms of kind of some of the other schemes that we've got at the moment, so we've got pilot projects and major projects, and early career researchers can apply as lead applicants for these. You just need to make sure you have someone that's got a kind of permanent position as a co-applicant, but there are rules around kind of whether you can apply for your salary or not. So make sure you kind of check that you meet the criteria and know what you're applying for. But one thing I wanted to flag was we've got all these schemes. We've made some changes recently in terms of like how we review grants, and kind of what the applications forms look like, how we can make sure we've reintroduced having people observe our grant review board meeting where they're making the funding recommendations. But the big thing I wanted to say is that we want to hear from you as to kind of what your experiences are as an early career researcher. What are we doing that works and works really well for you? What are we missing? What doesn't work? What are the barriers? Like what we kind of need to know so that we can look at our schemes, look at our support and tailor it so that it actually supports you as a community. Based on feedback, one of the things we've recently changed is our review process. We had external written peer review, which was kind of adding to like the burden on the research community, but also it meant that from submitting an application, it took 8 months to get a funding decision, and that's way too long. So we've tidied up our review process. We've kind of chopped and changed a few things. So now for some of the I say smaller things like PhDs and pilot projects, they're now taking a couple of months from you submitting an application to you getting a funding decision. Some of the fellowships and bigger grants are taking a little bit longer, around 4 or 5 months, but still it's way better than 8 months. And it means that you can get through that process and either you're successful, or if you're not, it means you can get back out and get back applying for things. We've also, following our pilot, we've reintroduced narrative CVs. So again, kind of trying to move away from just the looking at publications and looking at those metrics we traditionally looked at, but being more of a kind of wholesome look at someone's career and what the impact that they're making in research. One final thing on funding, hopefully you're aware of our network centres, but if you're not, I encourage you to take a look and sign up. If you're kind of based in Oxford, that's the Thames Valley network, or there's a London network. We have 12 centres across the UK, so I think definitely over 3,000. It's probably more like 4,000 now. But they offer, as part of that those centres, they offer kind of travel grants, equipment grants, pump priming grants, and not just that, kind of networking events, collaboration events. So if you're interested, take a look at our website and you can find the contact details of the relevant centre to sign up to their kind of network, to their newsletter, and then you can find out more about all the opportunities that are part of those. They also do public engagement events. They also do work around involvement and also looking at kind of EDI activities. So there's definitely a lot going on there. So I just wanted to kind of talk about the team a little bit so that you can kind of put names to faces in some cases, but just kind of get a bit of an idea of who we are, how we work, so that you know who best to speak to. This was taken at a conference a couple of weeks ago, and it's not just the research team. There's a few more teams mixed in there, but this is kind of how our research team is split. It's roughly into kind of 3 categories. So I'm in charge of research funding. So that's all about the schemes I've just mentioned, you know, making sure we're funding. My job is to make sure we fund the highest quality and highest impactful research into dementia. And we also, once we've funded it, we don't just sort of say, here's your money, goodbye. It's making sure that people are kind of keeping on track with their projects, if we can support in any way, and then helping other teams to think about the impact that those projects are having. And also a big part of it is early career researchers and kind of research culture and that sort of work. Our strategic programs team are more interested in kind of those bigger, longer term initiatives. So you might have heard about Drug Discovery Alliance. I mentioned about the blood biomarker projects, Brains for Dementia Research that we fund with the Alzheimer Society. That kind of sits in this team. And this team also help with impact and kind of making sure we demonstrate what we're funding is making a difference. And then clinical research is kind of a new function for us, but it's all about, we know there are some specific challenges around clinical research. So this team are supporting our kind of clinical research program, you know, work with our policy team, work across the organisation to specifically support clinical research. We've then got our science communications team, and I feel like I'm missing a few faces here, but they're not here, so it's fine. There are lots of ways that SciComms want you to get involved and work with them. So if you're interested in doing a blog, if you're interested in filming like a day in the life in the lab, I think last year they were asking for photos of labs with Christmas decorations. So there's lots of things that they kind of want to know about from you and to work with you. So if you've got a paper you've published, if you're, you know, really keen to help us like do like myth-busting sessions, and, you know, or working with press and kind of helping to issue comments, those sorts of things, do get in touch with the team because they're really keen to hear from you. I wanted to mention patient and public involvement. Obviously, Cherie's going to talk about this a lot more, but we too are really kind of supportive of patient and public involvement, and it brings so many benefits to research in general. I'm probably preaching to the choir, but you know, you get different perspectives, you improve the quality of your research, you make your research more relevant, and we do involve people with lived experience of dementia in our review processes. So they will be looking at if you apply to us, they'll be looking at your lay summary. They'll be asking questions of you in a fellowship interview. They'll be discussing with the other members of the grant review board kind of your application and how good it is not. So it's really important that you involve people with lived experience, not just at that point, but throughout the whole of your kind of research journey. And how we do that is, so we support our funded researchers and kind of facilitate involvement with researchers in our lived experience group. As I mentioned about the lived experience review, it's not just about high quality research, it's high impact, you know, what's going to really make a difference and mean a lot to people affected by dementia. So that's really important. And then clinical research, you know, helping to make sure that you know, involvement is embedded in our clinical work, but helping support people to kind of recruit to trials and recruit to studies. So what else have we got? So we've got funding, we've got kind of the team and how we can support you. I mentioned about Brains for Dementia Research. As I said, that's something we fund with the Alzheimer Society. So that's where it provides kind of tissue and data and lots of other things that you can apply to use. And it's available. The tissue is kind of available on. It's not just people. It's got a wide range of conditions, so it's not just people with Alzheimer's disease. It's different diseases, and there's kind of controls. There's the unique part is the kind of the clinical data that goes along with it. So that's a resource there. Mentioned our Drug Discovery Alliance. This alliance of kind of 3 institutes aims to kind of bridge that gap between academic research and science with kind of drug development in pharmaceutical companies, so kind of bring those two together a bit closer. And so if you're a researcher interested in drug targets and want to collaborate, either chat to me or kind of get in touch with one of our institutes. And that's just a bit about kind of what they do. And I've whizzed through this. I've still got a few minutes left, but I've only got 2 slides left. So one is about our annual research conference. So hopefully we saw you there 2 weeks ago in Birmingham. We made a change to our programme this year to make sure that early career researcher talks and kind of sessions were more embedded in the main programme, but we did have our early careers day the day before the main conference, where we had Sarah and the team do some fantastic sessions for the group. But as I said, we've just had it, so that's why I've got a few more grey hairs. But we're already planning for Manchester next year. So if you don't have it in your diaries, put it in now. It's the I should have checked this, 24th and 25th of February next year. And you can register your interest on the website. And there's more details on the website about kind of the prizes that you can apply for if you submit an abstract. So we have the De Laszlo Prize, which is an early career research prize, and lots more things that we're looking to do. And last but not least, we offer sponsorship for meetings. It's called conference sponsorship, but it's maybe a bit of a misnomer. If you're organising an event, you know, that's looking to kind of boost networking, make more collaborations, communicate or disseminate research results, you can apply for up to £2,000 from us to support in the delivery of that meeting. You'd be surprised at how few people apply for this. So, and it's quite, I'm not going to say if you apply, you get it, but the review process is short. And yeah, just going to leave that there. But please do take a look if you're interested in running an event. Then please do get in touch with us. I realise there's one thing that I forgot to mention about on the kind of public involvement and engagement slides. We also have an Inspire fund, so that's a small pot of money that you can apply to kind of disseminate research or kind of research activities to underserved communities. Again, that round is now closed, but we'll be opening later in the year. So again, do take a look at our website. Please keep in touch with us on Twitter. We're now on Blue Sky as well, which is exciting, and a few other channels. And my last kind of plea to you is, please sign up to the e-newsletter that we have. It's every quarter. And there's lots more kind of deadline details, opportunities, all that sort of stuff. That's where you're gonna hear it first. So that's it. I'm gonna stop. Thank you.
So thank you. And again, if you can just save your questions, we will have a Q&A And I just want to mention the Inspire Fund, that it's a small award that you can use for public outreach. And I was very honoured to win that a few years ago. And we ran workshops in the community. And that has had a knock-on effect. We now can continue to do those because we built relationships through a small award. An amazing grant, the Inspire Fund. Okay, so I would now like to welcome Cherie Nyota, forgive me for getting that wrong, from the Alzheimer Society, and who will tell us a little bit more about opportunities for ECRs, but including the importance of PPIE.
Hi everyone, welcome. I'm Cherie. I'm the Senior Research Grants Officer at Alzheimer's Society. And yeah, as Sarah said, I'm going to be speaking about the opportunities for ECRs, kind of what we offer as Alzheimer's Society, and then touch on the importance for PPI as well. So I usually like to start my presentations with a little bit about who I am and why I work for the Alzheimer's Society. So as you can see, this is kind of centered around my granddad. He lived in Kenya, and we'd go visit him kind of every 2 years as I was growing up. The two photos on the left are kind of taken 10 years ago. You can see my brother a lot younger, and he's very smiley. And then this was taken last year, which was the last time I saw him. And one of the reasons that I work for Alzheimer's Society, and I started out kind of working within the research engagement team and kind of being part of that, letting people with lived experience have that voice, is because I just recognized whenever we went back to Kenya that we didn't have a voice when talking about my granddad's care. They didn't really take it very seriously, dementia in general. It's getting a lot better now, but back then, 10 years ago, when it was starting to kind of develop, it wasn't really that clear. So that's why I work for Alzheimer's Society. I also have a little bit of a research background, not as extreme as yours. I'm not as smart as all of you, but I use that to drive my passion in research, but also kind of work at the society as well. So I will touch a little bit on our funding priorities. So one of the main aspects is we want to be the funder of choice for early career researchers. And that's so that we're increasing the number of dementia researchers across the pipeline. And I'll talk a little bit more on that later. We want to be considered leaders in EDI, equality and inclusivity. One of the things we're really looking at is our demographics, who we fund, and whether we are opening up to all dementia researchers. We also really welcome researchers from different disciplines coming in as well. That's one of the things that we're trying to kind of increase more of in our funding. Of course, we fund research of the highest quality. That's what my team kind of takes care of. And last but not least, we really want to ensure that people affected by dementia are key in the decision making. They're involved at every single stage, at that review stage, and then also post-award as well. So, as you all know, why do we invest in early career researchers? Again, it's drastically underfunded. We want to be increasing that research career pipeline. And we're investing in the future. We want to kind of build and maintain research capacity in the UK. So a little bit on our grants. So we actually are currently open for funding. We opened kind of end of January. We're open until the 28th of March. So not that long, but we have several grants, and kind of the ones that aren't blurred out, which is project grants, are for ECRs. I can go into all of these, but there's, as Jacqui said, we have lots more information kind of on our website as well. And you can come and ask me questions about any of these grants. I'll be here for the next 2 days of the conference, so please do come to me. But we do grants across the career pipeline. The only stage that we don't have grants for at the moment is PhDs, because we recently launched our DTCs, our doctoral training centres, so we don't have single PhDs, but we do have career development grants, postdocs, and those senior researchers of dementia research leaders as well. One thing that I would say is we recently ran 2 webinars on our process, on our outline proposal process, and that one of the webinars was on our new initiative that we put in, which is narrative CVs, and we had 3 members of our board, the chair of our board and 2 other members of our board, speaking about what you should put in your narrative CVs to capture their attention and make it engaging. So I would really recommend kind of emailing me. I'll put emails at the end, or come talk to me if you want a copy of that and you didn't get to see it, because it's a really, really good webinar, and I think it helps with my career development as well. So what are the opportunities we have for ECRs at Alzheimer's Society? So I'll go into the ones in bold and the other ones a little bit later. So one of the things is involvement in our grant rounds as expert reviewers and subject matter experts. So we have our regular grant round, and the way that we do peer review is we have what we call expert review panels, which are meetings with subject matter experts. And we always want to involve early career researchers in that. And we always go and seek from either our funded research and also our other externally funded researchers as well. And we also have those opportunities to observe our grant board meetings. All of our funded ECRs kind of get that opportunity, and we kind of go out in sections for different schemes that we do as well. We as a charity have dementia support services, and so across the country we have ECRs being involved in kind of talking about research at those support services. A lot of our services colleagues, kind of our operations colleagues, they're always asking us, can you get a researcher to come in and talk to a group that I lead? And that's something that we always offer to our ECRs as well. And then policy team, as you all know, there's so much going on in terms of policy, especially with these new treatments into dementia. So one of the things that the policy team is looking for is ECRs to come and speak with policy providers, etc. And so that's one of the opportunities we have as well. So we have a brilliant research communications team. On the top, you can see that's a grab from one of our webinars where Amy, who is our research comms officer, was talking about one of our dementia research leaders, Brian West. And then that's a blog from one of our PhD students about, and I didn't realize right now, they're both frontotemporal dementia. That's actually quite funny. So for our research communication team, they support researchers in media engagement, and they help to highlight their work. So as you can see, there's blogs and then our research webinars, and we also have lots of social media campaigns as well, and ECRs kind of drive a lot of that work. And research drives over 50% of our donations. So we really have ECRs involved in a lot of our fundraising opportunities, so speaking at legacy events, presenting at internal conferences for high-value donors and supporters, and then, engagement in our internal staff conferences as well. We also have our innovation team. This is kind of a new area for innovation. They've started kind of using our ECRs a lot more. I think one of the ways that ECRs can get involved with the innovation team now currently is collaborating on designing and co-designing kind of some of the innovations that they already have well established. They're looking for researchers to come in, look at those innovations and talk to them about it. And then there's also kind of this launch pad, which was recently launched, and this was to help early stage innovators test ideas and build prototypes. And one of the things that we'd really like to start seeing is if researchers have kind of research-driven innovations to come in through that launchpad scheme. And the innovation team is really there to kind of help build those ideas and take them from kind of research into practice as well. So our ECR retreat is kind of one of our biggest conferences every year. So you can see kind of some of the photos here. And we've run it for, this will be our 4th year running it. It's a 2-day conference. This year, it's in July, and I'll put up the dates in a second. But what we really focus on at these ECR retreats is career development and networking opportunities. So as you can see, kind of we have different sessions, such as on the top left, we've got the mock grant board. That's an opportunity for early career researchers to sit, and the grants team run what is close to basically a real grants board. So what we do is we get applications, real applications that have been applied to us. We get consent from people that have applied to us previously, and you are given those applications and you get to review them in advance, and then you have a discussion. And actually, one of our dementia research leaders sat on one of these. He's a fellow with us, and now he's on our grants board because we saw that kind of progression. So it's a really, really good opportunity. We have panel sessions as well. Soapbox Science is quite popular. It's where you have to do kind of a 3-minute talk really on your research and make it engaging and lay friendly. And we have judges for that. And we have different talks as well. one of the key things for us is networking. We want to increase the opportunities for early career researchers to speak to people from different disciplines. One of the things we're doing this year that's going to be a bit different is the app will have kind of a networking opportunity with business cards, and we really want to encourage that communication with each other after the retreat as well, like increasing those collaborations as well. And last but not least, our research network volunteers. So they are kind of at the heart of everything we do, and they are at our retreat as well. They will be running a workshop this year as well about patient and public involvement and using lay-friendly language within your kind of when speaking about your research. So they're honestly the reason that we do what we do. It's only because of them that we're able to kind of drive this research. So if I go into our research network, there's about 180 volunteers, and they're all experts by experience. So they either are living with dementia or are former carers or current carers, and they help us decide what research we fund and kind of ensure that research is impactful for everyone. Why we involve people with lived experience, it provides that different perspective so that we're improving the quality of research. It's making research much more relevant and supporting participation as well, because if we are involving people, that means they're talking about the research, and it increases participation in that research as well. Improving the accessibility of your research, so ensuring that, you know, the research is for those people with lived experience and it makes sense for them and it would work for them as well. Enriching and as I said, improving quality and increasing the motivation of the researcher. And I have a lovely quote later on about that as well. So when to involve, this is a question that we get quite a lot from researchers, is when to involve people in the research. And I think one of the things that we've noticed is that people will have the opportunity to involve when they're like forming their idea, but then they don't, and then that is reflected in that application. So one of the things that we do at Alzheimer's Society before the application start is we have this, you can apply to the research network to have a look at your lay proposal and see if it is lay friendly, have a discussion about your research to see if it's geared towards lay audiences as well. And that's something that really helps. We've seen kind of if someone has done that, we've seen them progress through the application stages and get funded. So one of the things I would say is if you are struggling with kind of writing your lay proposal, we do have that opportunity for you to email the research network team, the involvement team, and get kind of that lived experience on view on your lay proposal and see if that will help as well. But when to involve, you should be involving at every single stage of your research. So when you're identifying and prioritizing your idea, I think one of the things is, sometimes there's that thought that if it's like too biomedical science heavy, too basic science, you can't be involving people with lived experience. But that is involvement. That's using your, being able to communicate your science in a lay-friendly way. So if you want to discuss your applications with any research groups, we've got our research network, but there are local support networks across the UK where you can go and talk to people with lived experience about your research. And then at the deciding and managing stage as well, that kind of links into the identifying and prioritizing during your research. So what we do at the Society is all of our funded researchers meet with our research network volunteers once a year to discuss the applications, to discuss their projects. So they will meet with them, they will give that kind of lay perspective. If they have any kind of dissemination thoughts, or they will share that with the we call them monitors. They'll share them with the monitors. And we found that really helps kind of keep the researchers grounded, keep them thinking about exactly what they're doing this for as well. But also the research network volunteers, the people with lived experience, they really enjoy getting to learn more about the research. They review about 100 applications, 200 applications at the beginning, and they never get to, so if we didn't let them do this, they wouldn't get to see kind of the impact of that research, the funded as they go, and they really enjoy that as well. And then obviously at disseminating and that kind of evaluating impact stage as well. So what do our volunteers think? So I'll just read these out. So it's been a fantastic opportunity to meet with and get to know some of the most eminent researchers in dementia, and to have the opportunity to question their thinking behind research projects, and to hear them acknowledge the important role played by lay perspectives, the reality check. And this is from a lay reviewer. I carefully read all the research submissions and have enjoyed asking research teams questions to help improve or clarify their final submissions, as well as ranking their likely impact in relation to other proposals. So I just put this here to kind of really show that we do have them involved at every single stage. And because of that, I think one of the things that I really want to stress is, when you're creating your lay proposal to us, really talk about it to people that aren't within the scientific field. Like, talk about it with your mums, your dads, your sisters, your friends, your brothers, all of that, and give them your lay proposal to read and see if that is actually lay friendly, because they are judging it and they score those applications as well, and their scores impact what we fund. So I would really encourage kind of that involvement with your lay proposals. So if you want to contact us, we've got kind of grant inquiries is where you can come to us for anything to do with the grants. So if you have any questions about any of the outline proposal, your application, please forward it to grants. Or if you have a question for me directly, just come talk to me. The research network address, that's for anything to do with the involvement team. So they will answer any questions about that. If you want to have any kind of development with your lay proposal, that's who to go to. If you want to keep up to date with anything Alzheimer's Society related, we've got our Twitter and our Blue Sky. And if you want to join us at the ECR retreat, this is the link. Our registrations are open. We're seeing kind of a very large interest in those registrations, so please do register as soon as possible if you're interested. It's open to our funded researchers and also non-funded researchers by us, so please, we'd encourage everyone to come. It's on the 8th and 9th of July in Northampton as well. So yeah, thanks everyone.
I'm Squires and I manage the DPUK data portal, and a lot of work has gone into creating this really rich repository of data that early career researchers can access free at the point of use. So the data that's in it is quite impressive for the amount that you need to secure via funding to be able to access. I'm just going to go through the work that's been done over the last few years and where the portal is at the moment. Okay, so I'm just going to start with a quick snapshot overview of the data portal. So the data portal, as Sarah mentioned, is one of the arms of the bigger DPUK project. And it's been running for about 10 years now. We're in our second round of funding, and we have really been pushing to develop a multimodal rich data repository. We have over 85 data sets inside the data portal, and these have been collected from about 54 cohort studies. So lots and lots of data. We've had over 560 project applications over that period of time. So we have a lot of people applying to access this data and coming to us with their research projects to be able to do so. We've also, it's very global. We have applications coming in from all over the world. So it's up to the data providers to allow the access to that thing, but we will accept an application from anywhere to be able to open up this depository of data. And lots and lots of researchers have been using the portal. It's over 1,800 people have passed through DPUK in that 10 years as well. So we've been able to refine the offering that we have inside the data portal via those researchers. And the portal is a trusted research environment. And all that means is that it's a really secure analysis environment platform. And it means that we can hold very secure and private data and allow researchers to access that data, because we anonymise the data before researchers access it, and we make sure that the data stays inside that TRE at all times. So data providers will upload their data into that portal, and we hold them on secure servers inside Swansea University on our SERP infrastructure, which is ISO 27001 accredited, DAA accredited. It's just a very secure environment that's highly regulated. And it means that data providers are confident that we can hold that data and keep it private. And it means we can provide access to data that would be difficult to obtain access to otherwise. We then accept applications to access that data, and if an application is approved, we allow researchers to access that data via a virtual desktop environment. So they're never touching the data, they're only ever using a virtual environment to be able to access the data that's always on Swansea University service. And once the research has been done, researchers can apply to file out the data. So not the data, we would not allow that, the results. And so we're only ever letting out the aggregated results that have been generated, never any row-level data as well. And our trusted research environment, like many others, abide to the 5 safes, which were generated by, I think, ONS and has been further adapted by HDI UK, that lay out the requirements to be able to carry out a trusted research environment. And one of those things is we do a check on researchers coming into the portal to make sure they are carrying out, they are researchers and they belong to an organisation. And all researchers have to sign a data access agreement that says they're going to, behave in the portal and they're going to keep that data confidential and things. We also, when the applications come in, we do a triage of that application to make sure this is in public interest and that it's, ethical. And like I said, we always use safe settings, so that data is always inside that secure environment. We also filter the data that's given to the researchers. We only ever give out the data that's required to carry out the project that's been applied for. And like I said, we do this real output check-in. So 2 data analysts in our team will review every file-out request to make sure that no row-level data is going out of the environment. And we've really had, like I said, a real focus on multimodal data and bringing together not only the phenotypic data, but the imaging, neuroimaging, wearable data. There's all sorts of data that have come into the portal over the last 4 or 5 years now that has really enriched the data offering inside DPUK. And we've invested a lot of time and energy into data discovery tools. So Oxford University, they do the data curation work, which means that we can categorise and standardise and harmonise that data to be able to make sure that researchers can easily find the participants for their research as well. And as we brought in neuroimaging and genomic data, the size of that data increased and we needed to make sure that our compute environment, so there's a HPC cluster inside our trusted research environment that can be accessed by researchers on a cost recovery basis. But that means that you don't have to take the data out anywhere. You can do all your analysis inside that environment. This is just a quick example of our data discovery tools and the matrix showing the categorisation level carried out by the Oxford team for the data portal. And we also then allow researchers to go down to the variable level, and they can also go deeper again using our Explorer tools, which are all on our website and can be accessed. Like I said, we have Windows desktop virtual desktops, and we also have Linux virtual desktops, because particularly with neuroimaging data, a lot of the software that needs to be used to analyse neuroimaging data require a Linux environment. So that's something we had to spin up quite quickly to meet our researcher needs. So even if we're not doing what you need to be able to carry out your research, or you need particular or specific software, we can work with you to make sure that we can enable that in the part thing. But our virtual desktops come as standard with all the software you see on the screen and more. Our new engine pipeline, so we have quite a big team that work on this to make sure that we can bring in neuroimaging data, that it is anonymised, it's defaced, it's curated, and we also pre-process that data as well to generate IDPs, so that researchers can apply to just access the brain volume measurements, etc., alongside the phenotypic data, and it reduces the skill set required to be able to access neuroimaging data. We also have a lot of work on genomics data as well. So there's a study carried out between Oxford University and Cardiff University, and we're adapting it in Swansea University to make sure those pipelines are available inside the portal. And this is to be able to generate polygenic risk scores from the cohort data that is available. And there is polygenic risk scores now available for 6 of those cohort data sets. So you can see that as we're going through these funding periods, we're trying to enrich the data as much as we can for the researchers to be able to come in and to be able to use that. We're also looking very much at federation. At the moment, you can come to DPUK and you can use the data in DPUK, but what if your the data that you're interested in is in DPUK and another TRE? Well, at this moment in time, you'd have to go to both TREs and carry out your research and bring them together at the end. We're looking at having a federated network of TREs where you can submit a query and that query goes out to all the TREs and the results are returned to the central node. And we're also looking at popping up a TRE governance, tightly governed TRE in between other TREs. So you can work inside your usual environment, but actually you're able to access data from multiple TREs. And I think this would be really important when you're starting to look at AI models and having enough data to be able to train those models. There's a lot of work that goes on the registers and trials from the Oxford side as well, bringing in the participants to be able to create a register that can be used by researchers to select participants for their own studies as well. And a lot of enrichment of the data happens through these registers in, collecting wearable data. There's ApoE status collected for all the participants. So this is another area that we're pushing forward in. And we've done a lot around artificial intelligence. There's lots of applications that are coming through to DPUK with an interest in developing AI models. And this posed a real problem for us because there's always a risk that they could leak data if they were released from the environment. So my colleague Lewis has led on a big piece of work to be able to ensure that we have the frameworks in place to be able to allow researchers to come in and develop those models, and we're looking at options to be able to host those models further on, how we can share those models to other academic institutions. And I think we're one of very few TREs that are able to accommodate AI model release and the discussion around that. So it's really important work that's been carried out. We're also doing a lot of work on synthetic data to be able to allow researchers to be able to access synthetic data at a lower level of governance. So you don't have to go through all the triaging. You can actually go and explore the data, better data discovery tools, because you can actually go and see the data because it's synthetic, but it is representative of the data inside the portal. So that's ongoing, and also generating synthetic data to provide as controls or to supplement the data that we've got for AI model training. So, we've invested a lot in that as well. And the last thing we're looking at as well is large language models for data discovery. At the moment, we've got really good data discovery tools, and I think the Oxford team has ensured that we've had the curated data to be able to do those, but it would be great to start having a more interactive, large language model-led discovery tools on our platform. So my colleague Abdul and Lewis are doing a big piece of work on being able to do that, and they're around if anyone wants to chat about that as well. I've just got this last little slide. This is the data pool. There's a lot of people involved in making sure that data is fit for researcher access. So this is the Oxford and the Swansea team, and the work that they do to make sure that data is available and researchable is fantastic. And they're a fantastic team as well. And that's the end of my chat.
I really want to just cover really quickly some of the opportunities that DPUK has developed over the last 7 to 8 years, but also some of the training opportunities that we want to roll out next year in 2026. Now, a lot of our training opportunities have been developed after recognizing the need of researchers. And mainly the need is that we found researchers just don't have affordable training. And what we wanted to do at DPUK is develop cost-efficient training opportunities for researchers, but also research training that is focused on large cohort data sets. And so that's what we really set out to do a few years ago. And we find that researchers come to use the data portal, and it's a little bit like a candy shop. They see all the data sets beautifully laid out by the Swansea team in the metadata tools, and they click tick, 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 10 data sets. And then a year later, they actually haven't done anything because it's overwhelming and they don't know where to start. So really the aim of our training academies is to help with that launching. How do you work with multiple data sets? How do you harmonize? Do you have to harmonize? Do you standardize? And where do you start? And so that's really the ethos behind the training academies and datathons that we provide. And during the pandemic, we took them online, obviously, but we've left them online because we've realized that they have global reach. So we have researchers from over 30 countries, South Africa, Colombia, the States, Australia. And so that's why we keep them online. So just a little bit about our training that we offer. Our academies, we have an elementary academy in the spring and an autumn academy, which is an advanced academy. Now, the spring academy really assumes no experience whatsoever. It can be used as a refresher course for those who have only done undergrad statistics. It's conducted in R. We work very closely with Edinburgh University team who conduct this over 5 days online. It's basic statistics. It's ideal for students, but also for clinicians who've perhaps gone the medical route, but now want to do a research module within their medical studies. And it's really, really affordable. It's £125 for 5 days. We don't take a profit. We give all the money for our lecturers as honorariums. And our next one is the 7th to the 11th of April, 2025. So coming up. So if you are interested in this academy, or you know people, colleagues at your institutions, please do sign up fast because that deadline is approaching. Then the Advanced Academy, this is really for experienced researchers. It builds on the Elementary Academy and plus. So it's conducted in R and Python. It's longitudinal statistics. So we cover machine learning, longitudinal structural equation modeling, Cox regressions, survival analyses. So it does, we do look at CVs. We do make sure that our attendees are experienced enough to cope with the 5 days. And again, this is hugely popular, and these are both on our website. And if you're interested, do sign up, or there is a link, I think, for an expression of interest. And our next one is the 3rd to the 7th of November. So for both of these, there is not a limit to the number of people that can attend. So whether we have 30, 50 or 100, there is no limit because we do run these online. Now, the one that is incredibly popular that does have a limit are the datathons. These are run in June, and these are, again, we took them online during the pandemic. It's a 5-day workshop, and we come together in groups. So you apply, we only take 30 individuals for this across institutions at varying levels of expertise. We group everybody up so that you are spread across expertise. It's aimed at ECRs who want to make new collaborations and who are interested in taking beginning analyses forward to a publication. So I provide mentoring support from Oxford for 12 weeks. We work closely with the Swansea University team who provide the DPUK virtual desktops for 12 weeks. And our next datathon is the 16th to the 20th of June. Now, this is free because we don't provide teaching. This is our next slide will show you. The theme for this year is a multi-cohort approach to dementia research. We have 6 of our cohorts who work with us, so you have access to these 6 cohorts. We group you up. over 5 days. You interrogate the data online. You see what you can do over 5 days. We have a presentation session on the 5th day. I mean, this is when we had it in person, hugely popular. So if you are interested in this, I do suggest you sign up, because we are usually heavily oversubscribed for this opportunity. Now, coming in 2026, we are going to roll out one-day workshops, because we do appreciate not everyone can take off 5 days for training. And the workshops that we are launching, and this is based on feedback from individuals that they would like these training, is structural equation modeling in Stata for beginners. For those of you who are familiar with Stata, it's a fantastic package, and it's really great for doing structural equation modeling. So there'll be one for beginners. There'll also be a more advanced one on longitudinal structural equation modeling, so cross-lag panel models, latent growth curve modeling. Again, this will be a free one-day workshop, and it will be held online. And then multi-cohort data management for researchers. What we find is that simply understanding where to start when you have 5 cohorts, what do you do, where do you start, is often quite complex and daunting. And then finally, multimodal data modeling. How do you bring together clinical survey, some imaging and phenotypes and genomic data into that one model? How do you work across the desktops that Emma mentioned, such as a Linux desktop and the other virtual desktop environments? So these will be launched next year, and the dates will be put up on our website. Now, another initiative that we are launching in collaboration with the International Health Cohorts Consortium, on which I am a member of the steering group. If you are a genomic analyst, I do suggest you look the IHCC up. They are a big organization that initially was launched for all cohorts above a population of 100,000 individuals. They've now softened that rule to more longitudinal populations with a focus on genomics. And what we're doing between the two platforms is we're launching this initiative. We've run a pilot study over the last year with 2 researchers, where a researcher, you identify a mentor yourself, and you come to us. At the moment, it's just me. So you come to me with the name of your mentor, and you connect with the DPUK training sessions. So that's the autumn and the elementary academy and the datathon for free as part of the RMP. And you meet with me and your mentor once a month, and you run a project through the DPUK data portal. So you submit a project proposal, but I work with your mentor to have a project completed in 1 year. So we're limiting it at the moment for only 4 researchers in the first year. The pilot project was very successful. So if you are interested in being part of this first year cohort, there'll be 4 researchers in the April intake and 4 in the November intake. I'm talking without my slides. I always do this. So basically, this will be the process. You identify your mentor, you scope out your project, you submit your proposal as if you were just a researcher applying to DPUK. It's reviewed in the normal way. You gain your access, you join the training programs, but in the background, you are meeting with your mentor and a member of the DPUK team, which is me for the first year. And the benefits of this process for researchers is that you get mentoring from a bespoke person in a structured format with a structured project through DPUK. You get experience of being a PI on a project for the so for some people, for the first time, you get a publishable output. That is one of the objectives of this RMP, and career guidance from both your senior mentor and the person from DPUK. And for funders, this is very favourable as well. If you're going for a fellowship or a small grant, this looks good. If you say that you're already working with a mentor, you have an established project ongoing through DPUK, this really does look good. If you say that you are registered in an ongoing training program. So please do email me if you're interested in this first year that we're launching it from April, so that we can sign up the first 4 people for running this. So I hope these are of interest to you, and that they will serve gaps in the training community. Thank you.
Hi everyone, so I'm Chelsea and I'm one of the translational and clinical scientists at Alchemab. I'm going to talk to you about the development of a novel biomarker assay and then subsequent data that was produced from multiple neuropathology cohorts which have Netrin-1 linked cognitive impairment. So Alchemab works slightly differently to traditional drug discovery. We start with patients rather than a target. We hope to reverse engineer the adaptive immunity process to discover therapies that have come from autoantibodies, and then we can use these to treat patients. So our bioinformatics team started with over 13 million sequences from B-cell repertoires, and from this they then came out with 68 potential therapeutic antibodies. This was prior to sending it to the research team for screening. Now, as I said, we don't start with the target, so we have to deconvolute. And for this, we use protein microarrays. We found that the target for these antibodies was UNC5C. Now, this is a receptor of Netrin-1, and Netrin-1 is very important in axonal guidance. We also perform cell binding assays to make sure we have endogenous function, as well as looking in receptor homologs to make sure there's no off-target binding. We performed X-ray crystallography with the protein sciences team, and they look at the receptor ligand complex. And with this, they looked for UNC5C and Netrin-1, and then also UNC5C with our antibody. And we discovered that they had a competitive binding for the same domain. So then we performed a phage campaign to look for other antibodies, hoping that we could create an assay that wouldn't be impacted by potential therapeutic antibodies in later patient samples. We optimized the assay through a series of DOA experiments and transferred it to MSD, which increased the dynamic range and reduced matrix effect. You'll also see that there's really good cross-reactivity in relevant samples for human, mouse, and cyno, which will be important in clinical studies later. Now, I said that this came from an FTD cohort originally, and FTD is a group of different dementias that result from neuronal loss of specific areas in the brain. This commonly has impact on speech, behavioural changes, and executive functions. It's also one of the dementias which affects people earlier on in life, and there's a typical age of onset of 56 years, with an expected life expectancy of 7.5 years post-diagnosis. Now, when we looked in literature, there wasn't a direct link between FTD and the Netrin-1 and UNC5C pathway. But what we did see was mention of axonal guidance cues and apoptosis. So this is really important in that Netrin-1 pathway and really important in FTD. So we looked for other neuropathologies that might have a link to this pathway. And most predominantly, we saw Alzheimer's disease, but there was also links to Parkinson's and ALS. We also looked in mass spec data sets of relevant samples. We knew we wanted to look in serum and bloods. And we only saw that there was extracellular domains present. And we thought this must be from maybe some form of shedding rather than neuronal damage, because otherwise we would expect to see that there would be domains throughout the entire of the protein. So fortunately at Alchemab, we have some great collaborations, and we were able to look across multiple neuropathology sets, as well as lots of healthy subjects, and we also included rheumatoid arthritis. This was going to be a non-neuronal inflammatory control. And interestingly, what we saw was elevation of soluble UNC5C was isolated to just neurodegenerative disease cohorts, but also we could detect it in some really early sets. So we included preclinical AD, where there's increase in amyloid and tau, but no cognitive impairment seen. And then also this prodromal cohort, which is rapid eye movement behavioural disorder, and this commonly progresses to from a form of Parkinson's. And really interestingly, what we see here is that within prodromal, there's actually most elevated level of UNC5C, and we think this is because there's active neurodegeneration occurring prior to that progression into Parkinson's. I've also highlighted ALS, so this was our largest cohort, and then we looked further into this cohort. So within Alchemab, we stratify our patients into different resiliency cohorts. And with this, we generally have resilient, semi-resilient, and progressor, but also here we have unknown. This is because there's probably too few data points to either determine slope rate. We also factor in things such as age of onset and other biomarker data that comes with these samples. And what we really were encouraged to see was that between control and resilient patients, there wasn't a difference in the levels of UNC5C that were seen. And this links back to that idea of protective autoantibodies. And then we also saw that in semi-resilient patients and progressors, there was an increase in this UNC5C. And then also in the semi-resilient, when you take back that look of prodromal compared to Parkinson's, there's a higher increase than the progressors. So this could be a sign of ongoing neuronal damage at this current stage. So what we set out to achieve was a target engagement biomarker assay. But what we think we've managed to develop is an assay that could be used as a diagnostic or prognostic biomarker assay because it can detect elevation of soluble UNC5C earlier on in disease. I'd like to say a massive thank you to everyone at Alchemab because this has been a huge collaborative effort, and also all of the biobanks and especially all of the sample contributors, because without them we wouldn't have this data.
I'm Meggie Smith, a DPhil student at the University of Oxford, and today I'm going to be sharing with you some findings from my study looking at the mind, diet and cognitive performance and whether this relationship is moderated at all by the presence of cardiovascular risk factors. So as a way of a very brief introduction, we can see from the literature that diet can influence brain health and cognitive performance. The mind dietary pattern is a dietary pattern that was created specifically to support brain health and reduce the risk of cognitive decline and dementia. It was constructed based on the strongest findings in the fields of nutrition and dementia, but broadly it's a combination of the Mediterranean diet and the DASH diet, both of which are known for their overall health benefits. It's a whole dietary pattern and is composed of 15 components. 10 of which it promotes consumption of, as these are thought to be good for the brain, and 5 of which it states limitations, as these have been deemed unhealthy. A growing body of evidence indicates positive associations between consuming the mind diet and a range of cognitive outcomes. However, these positive associations have not been observed in all investigations. Additionally, key questions remain in terms of the longevity of these associations and whether certain groups are more responsive to the effects of this diet. Shared biological pathways between the cardiovascular system, the diet, and brain health suggests those at risk of cardiovascular disorder may be positioned to experience heightened cognitive benefits from following this diet. So building on all of this, our key questions to investigate were, does consuming the mind diet affect later life cognitive performance? And does the presence of cardiovascular risk factors modify this relationship at all? To address our questions, I used data from the longitudinal cohort study called EPIC Norfolk. This study measured dietary intake and the risk of cardiovascular disorder, and then around 11 and 23 years later, they measured cognitive performance. Using the dietary data, I scored how well each participant adhered to the mind dietary pattern, with scores ranging from a possible 0 to 15. To explore the long-term cognitive associations of consuming the mind diet and whether this varied based on the presence of risk factors, I conducted a series of structural equation models. First, I created latent constructs of cognitive performance at each time point using confirmatory factor analysis. This produced a construct of general cognitive ability based on the performance of 4 different tests. These constructs were then used in the following models. I then split the sample into higher and lower risk of developing cardiovascular disorder using a clinical measurement tool called the Q-RISK-2. I then performed multiple group structural equation models with Q-risk as the grouping variable, and this method performs the model separately for both groups, allowing exploration of any differences in the relationships. So these are the graphical representations of the analysis performed with the significant pathways highlighted in pink. Here, we found that in individuals with fewer cardiovascular risk factors, following the MIND diet was associated with better cognitive performance 11 years later. However, this positive association did not extend to cognitive performance 23 years later. Conversely, for those at a higher risk of developing cardiovascular disorder, following the MIND diet predicted better cognitive performance 11 years and 23 years later. So together we found that the impact of following this diet on cognitive performance over time depended on cardiovascular risk level. So to conclude, diet was associated with cognitive performance many years later. Our results suggest there may be cognitive benefits to everyone following this diet, but individuals at a higher risk of cardiovascular disorder may experience longer-lasting cognitive benefits. Together, these highlight the importance of dietary habits for cognitive health, particularly for those with greater health vulnerabilities. Future studies should continue to investigate how diet can optimize cognition, particularly exploring whether certain groups maximally benefit from certain diets. Additionally, whether there are key time points where diet has the greatest impact on later life cognition should start to be explored.
How fantastic to be here. Dementia's Platform UK Translation Conference. So are you having fun? I can't hear you. Are you having fun? Come on, are you having fun? Yeah. Scientists and clinicians, I guess. So I think I'm here to talk about my career. I'm Atticus Hainsworth. I'm a neuroscientist. I work at St George's Hospital in southwest London. I've actually had a very checkered career, which I'm not going to go deeply into. I think my only claim to fame is persistence. I have actually kept going. I kept sort of shoveling along, and I'm very happy with how my career's gone. I've got a very nice wife and nice kids, and I get a decent paycheck, and so on. So that's my career. But I actually wanted to say two other things to you. As I think predominantly early career researchers. The first is there's a danger you might feel as you come into this kind of research career path. I'm such a little guy. I'm such a small fish in a big pond. It doesn't too much matter what I do. No one's noticing. That's not true. They are. It behoves us at all times to behave in a professional manner. We are professionals. And in fact, we represent the discipline. We're dementia research. To the person you talk to on the train going home tonight, you're representing us as a field. And by the way, in 10 years' time, you won't be the little guy anymore. You'll be reviewing my grant if I'm still working, which I might not be. So you're at this fantastic conference. The team have done a wonderful job putting the program together. You're listening to giants in the field. John O'Brien, Donna Wilcock, flown in from Indiana, Paresh Malhotra. These are the names in dementia research. But you know what? They're going to retire as well. And who's going to fill that gap? It's got to be you. The next thought leaders are in this room. And we very much want to keep you in the field. Dementia research needs the smart and the creative and the slightly disruptive dementia thinker, research thinker. So that was my first message. You do matter. Of course you do. You knew that anyway, but I'm just underlining it. My second point, I suspect a lot of you are in the academic part of research. There may be some industry people. You may go into industry, but we've sort of done a PhD and gone into university life because we're not entrepreneurial. We're not there to make a our first million in the next 5 years. We love the research. We love the test tubes and the lab and the patients and so on. Actually, I would put it to you, and if I could, I would have said this to my 25-year-old self. Yeah, you are an entrepreneur. You're selling something. And that something is you and your skills. You have something to sell. When you write a grant, you'll realize you're selling something. You've got to make the case that grabs the funder such that they say, well, okay, we're going to fund her, not him, because she's made the case. She's sold the idea to us. And there's a hell of a lot of that in the research life. You're not just a super brain in your ivory tower or your lab slaving away at sort of transcription factor number 4. You have got something to sell that matters societally. So we did my career, we've done my 2 points, and I think my 5 minutes is probably up. Thanks very much.
I will just briefly talk you through my career. So I'm Simon Young. I'm Senior Programme Manager at Dementias Platform UK, based at the University of Oxford. Many, many moons ago, I did a PhD in statistics. And then I decided not to go down the kind of route that Atticus was just talking about. I decided to go into industry, and I worked as a statistician in the chemical industry for a period of time. After that, I moved over into insurance and worked as a statistician in the insurance industry. And there's a bit of a theme coming on. I got a little bit bored of that and then moved into a different industry. I worked as a statistician in the credit industry. Then I developed out of statistics. I still do a little bit now and again, but moved more into general management and project management within that credit industry. And then I thought, well, no, maybe I'll do my own thing. I'll have my own company. So I formed my own company, did some consultancy. And that worked well for a while, but I got offered another role back into management. So I thought, well, I'm not going to turn that down. I'll go do that for a little while. But then I remembered why I went back into independent and moved back out into my own consultancy company again. And within the previous part, I'd had experience of different areas, and I became a main board director for a company in the UK and also for a company in Spain. But I was still finding there were different things that I was interested in, and moved into independent consultancy yet again. And within that, the opportunity came up to move to the University of Oxford and work with John, Sarah, and the team in Dementias Platform UK. And I've been working in that area for just over 6 years now. Time has flown past. And thoroughly enjoyed my time there. So, as you can see, I've done quite a lot of different things. And I suppose that is the message. You don't have to just go down one track. You can do what you want to do. You can move around, and I believe moving around is becoming more and more popular, more common as we develop. I would just like to share with you a few things that I've learned and observed over my career. And the first thing and the key thing, and I think it's sort of what Atticus was talking about as well, is do what you enjoy. You've got to work out what you want to do. And that is what you want to do. There is nothing worse than dreading going to work and doing something that you don't want to do. So find out what you want to do and then go do it. But there's not a problem in changing your mind and trying to do something different after a while. In fact, there are many benefits in doing that sometimes. There is no correct answer. There is no correct standard career trajectory. You don't have to go into the lab and do the test tubes and then move on to something else. It depends very much on what you want to do and what is right for you. So don't feel constrained. There is an issue that the grass is always greener. A company comes to you and says, I'll offer you this huge amount of money to do this wonderful work. And you think, wow, that sounds excellent. And it may be, but it may come with other things attached to it. So there is a danger that what you're working on at the moment sounds ordinary and what you're being offered sounds wonderful. You've got to bear in mind that life's not always quite like that. Not what you're being promised isn't necessarily what you're going to get. I would encourage you all to try hard. Through my career, I've observed that the people that try the hardest are usually the luckiest people. Things work out for people that try hard and work hard. So when you've worked out what you want to do, go for it, work hard on it. But also be kind. That's a general rule in life anyway, I suppose. But a lot of what makes it good for you and also good for who you're working for and who you're working with is to collaborate with people, to learn from other people that you're working with and to share your knowledge with those people. And people will always wish to work with people that they find kind and easy to work with. So, in my opinion, if you follow those rules, then I think you'll have a successful career. Thank you.
Thank you very much. Well, two great talks and two great bits of advice. So I've been asked to do an address, which is going to be a bit of a sort of ramble, combining a bit of history, a bit of Lewy body dementia, a bit of my career, although not too much of that, and most importantly, maybe some lessons learned both about the disease, but also about careers as well along the way. So that's kind of the text for today, if you like. So let's start with Terry Pratchett. People know Terry Pratchett, best-selling author, who unfortunately developed post-cortical atrophy and died a few years ago. And one of the things he wrote in one of his novels, which I think is so true, is about the importance of history. If you do not know where you've come from, you do not know where you are. If you don't know where you are, then you don't know where you're going. And if you don't know where you're going, you're probably going wrong. So I think in all that we do, we should just remember how have we got here, and we don't want to repeat mistakes of the past. Now, if we start with Lewy body dementia, we have to go back quite a long way to Munich in the early part of the 20th century. And there you had the most amazing set of career researchers. They weren't early at the time, but nonetheless, you had Alzheimer in the same lab as Friedrich Levy. And of course, he was pronounced Levy then because there's no W sign in German. And he described the Levy body, which is the characteristic feature of Parkinson's disease. But he later moved to the US and sort of Americanized his name, and he became Louis. And when I was at your stage in the audience and I went to sort of conferences like this, the biggest debate about Lewy body dementia was what you called it, whether it was Levy body dementia or Lewy body dementia. And people got very passionate about this, such that someone would talk about Lewy body dementia like I am. And at the panel discussion, someone from the audience would get up and just berate them for being so ignorant because they couldn't pronounce, the proper name, Levy. And there was a lot of passion and emotion, and actually there was more to it than the name. It was all to do with kind of socio-political Nazi regimes and all sorts of things, which I didn't understand at the time as an early career researcher, but there was a lot of baggage attached to whether you said Louis or Levy at that particular time. Things have fortunately moved on, but I suppose I say that partly because terminology and how you communicate and talk to each other is incredibly important. And it's been very important in Lewy body dementia because we've had so many different names and terms for it over the years. Dementia with Lewy bodies, Lewy body dementia, Alzheimer variant, senile dementia of Lewy body type, all these things. And it continues to be important. So we had a wonderful meeting in Amsterdam in January this year of the consortium, which I'll say a bit more about later. And the debate was all around dementia with Lewy bodies and Parkinson's disease dementia, these two conditions that we think of as two ends of the spectrum. And are they still useful concepts? So I think it's important that we think about language and we think about how we communicate. And it's really, really important to know what you're meaning when you describe things, because otherwise you can get great misunderstandings, and that's certainly been a feature, I think, of Lewy body dementia. But going back to Lewy body dementia, where's it sort of come from? Well, it was really described from case reports in the early 60s. And it was only when I got interested in it, several decades later, I sort of realized I'd kind of grown up with Lewy body dementia. It was actually first described before I was born, but nonetheless, I was a child of the 60s. I later discovered that Lewy body dementia was a child of the 60s as well. And it's just interesting to see how it's developed over that time. So there were case reports from the US, from Japan, from other places of people that had Lewy bodies in the cortex, and they didn't just have sort of motor disorder like Parkinson's disease. They had dementia as well, and people linked these and also noticed that people had hallucinations and other things. And this led to the formation of kind of case series, like from the Newcastle group, who played a great role in defining Lewy body dementia early on. Not the only group, of course, but one of the groups. Doing very painstaking studies, looking back at case notes of people who died that had these Lewy bodies in the cortex, and trying to say, well, was there anything different about these people during life that meant we could have recognized them? And they found that a lot of them had visual hallucinations, a lot of them had unexplained delirium or fluctuation, and a lot of them had Parkinsonism. And so this sort of led to the formation of early thoughts about diagnostic criteria for how you might sort of detect this during life. And also recognition that there were quite a few people that were suffering from this disorder, or had this disorder rather. And the question arose, well, is it new or is it old? We haven't recognized this before. We're suddenly seeing it in the 70s, 80s. Is it a new disorder? And there have been some very careful pathological studies that have been done by people like Blessed, Tomlinson and Roth that had been instrumental in showing that the main cause of dementia in later life was actually Alzheimer's disease. Because before these studies, most people thought that the main cause of dementia in later life was vascular, arteriosclerotic dementia, blood vessel problems. But they clearly showed the main cause of dementia in old age was similar to the Alzheimer's first case, which was described in a 55-year-old lady. Alzheimer's disease, it was the same plaque and tangle pathology. And so these were the sort of causes they found and they reported in the 70s. There's no Lewy body dementia there at all, of course. But nonetheless, that's a sort of perfect series to then go back and say, well, were there some cases that weren't recognised? And that's one thing that Robert Perry, neuropathologist, did. He actually went back to that Tomlinson series, same series of cases, now staining and looking for Lewy bodies. And there they were, 20% of cases. And that's very similar to the pathological series that you see now, some series even slightly higher than 20%, 25%. So I suppose that's one lesson in, historical data and revisiting it with new methodologies. And that was really informative because that simply told us that it wasn't a new disorder. It had been there all along and just wasn't recognised. It was known as something else. Around that time, there was an international consortium that was formed by my colleague Ian McKeith, who's sitting in the middle there. And I was pleased to be able to join that as an early career researcher, just as things were getting going. And I suppose I learned a number of things through my involvement with this consortium. One is that coming to meetings and conferences like this is absolutely fantastic. And it's really hard to put a value on it and articulate the value because the kind of networking, the interactions, what you gain and learn is tremendous. And in person is just so much better. So, online stuff, absolutely fine. But coming to meetings like this, networking is great. And the other thing is that, knowing key people and particularly having them as mentors or role models is key. And I mean, Ian McKeith has been a tremendous mentor and role model for me in terms of advice and in terms of guidance and opening doors and things. So I think that's really, really important to find people. And I've been fortunate to have several along the way. And that really, really helps. So, you can do things on your own, but, link up with people that you think are the role models and where you want to go to. And you'll find most researchers are very interested and receptive in that. And they want to help. Might be someone you work with, might be your line manager, might be somebody else, along the way. Though the criteria moved on for Lewy body dementia from sort of thinking about clinical features to actually international groups coming together and saying, yes, we agree these are the clinical criteria. We agree these are the pathological criteria. The details don't particularly matter, but I mean, you all know Lewy body dementia is dementia and then one or more of, fluctuation, hallucinations, Parkinsonism, REM sleep behavior disorder. You need one feature for possible, two features for probable, and that's pathological confirmation. So this was fantastic because you had a similar language that everybody could speak. Pathologists around the world knew, methods might vary, but there was a common language. So that's going back to sort of having this commonality, which really helps things move forwards. And it was around that time that biomarkers were developing. It's interesting because in many ways, biomarkers for Lewy body dementia came around the same time or even earlier in some ways than some of the ones for Alzheimer's. Though, of course, Alzheimer's has overtaken Lewy body by a long way. And there were two that I particularly worked with in my career over the years. One was almost the absence of a biomarker. So it was the fact that in Lewy body dementia, you have relative preservation of the medial temporal lobe, unlike Alzheimer's disease and frontotemporal dementia, where you get quite marked atrophy. And the other was loss of the dopaminergic system, just like you see in Parkinson's disease. It's a good biomarker there. And it was shown that it's a good biomarker also in Lewy body dementia of the Lewy bodies as well. And I was fortunate to come across this person, Philip Sheltons, and again, I met him at a conference. He was an emerging young neurologist. I was an emerging young old-age psychiatrist. And we met over coffee and we just talked, and I was interested in imaging and hippocampal change. He was interested in Lewy body dementia. We didn't know a lot about each other's fields, but we thought it would be interesting to explore things further. And that was the start of a great sort of friendship and collaboration over the years in terms of, imaging in Lewy body dementia. And Philip, as you may know, is now president of the World Dementia Council. So you could say his career's gone slightly better than mine, but it was great. And I think that's the other thing. It's finding people along the way, isn't it, that you get on with and you gel and you have similar way of thinking, and that can be really helpful. And a number of, you can reflect on people. So as well as sort of mentor, you think of someone senior as a role model, maybe someone who's a sort of contemporary as well in your journey along the way is incredibly important. Now, when we were looking at dopamine imaging, and this led to sort of licensing in the EU and a lot later licensing by the FDA and others as well, we found that the sensitivity of this method, I don't know how well you can see this, but in Parkinson's syndrome, call it Parkinson's disease for simplicity, the sensitivity of this method, which is, the absence of dopamine transporter binding in the striatum, was about 90% sensitive and 90% specific for Parkinson's disease. And it was much less when you looked at the pool data for dementia of the Lewy body. It was only 80%. So, 1 in 5 cases with dementia of the Lewy bodies were not being picked up by this biomarker. And all the tests, the validation that had been done to date had been on clinical diagnosis as the standard. So it wasn't quite clear whether this biomarker wasn't quite as accurate in dementia of the Lewy bodies as Parkinson's, or whether we had some misdiagnosed cases there. And it was a debate that went on, and there was no way of kind of resolving it without knowing, because we didn't have pathology at the time and we didn't have other biomarkers. So the way to answer that was indeed to get some pathology, and we set up this longitudinal study, and took ages. It took 10 years really to get data. But the thing with a study where you've got kind of imaging during life and pathology is you don't need many cases to maybe answer the question. And in our first series, we actually only had 2 cases. They're the red dots with the black circle round. And what those cases are cases that had normal dopamine imaging during life. That's why they're high up here on the y-axis. But at autopsy, which was many years after the scan, they still had reasonably preserved counts in the substantia nigra. In other words, their dopaminergic system was not very severely compromised. It was a bit compromised because there's a lower end of the kind of Alzheimer's spectrum. But nonetheless, they had ample evidence of cortical alpha-synuclein. So they met pathological criteria for dementia with Lewy bodies. They had that clinical syndrome during life, but the biomarker during life had been all normal. So, you know, only a couple of cases, we had definitive evidence that this biomarker didn't pick up everybody. And it wasn't just that you'd sort of misdiagnose people during life. And I suppose to me that was an example of a different sort of methodology, because I'd never really been involved in pathological studies before, of how that was really sort of important. So diagnosis of DLB has gone on, of course. Pleased to see it being taken up in DSM-5 and now ICD-11, and that's led to a lot more interest in the disease, because pharma in particular were always a bit cautious about dementia with Lewy bodies, because they said, well, it's not in the DSM and it's not really an ICD, so do people really recognise it as a disorder? But now it is, and I think one advantage is it means that people can now get a formal diagnosis through whatever system. And that's also led to formal inclusion in guidance. It's NICE guidelines 2018, which are the current ones that advocate, for example, use of dopaminergic imaging. So it's also for me an example of just how long things can take, because it's at least 20 years between the first demonstration of dopaminergic abnormalities as a biomarker for DLB and someone like NICE actually saying, yes, this is a clinical test we think you should do in the NHS if there's diagnostic uncertainty for dementia with Lewy bodies. So translation is possible, but it can take a bit of time. But if you stick with it, you do get there. Continuing a theme of diagnosis, because it's always been quite a challenging diagnosis, and DLB's always been sort of under-recognised. We did a program, the Diamond Lewy program, which looked at the diagnosis and management of DLB in the UK, just to see how many cases were currently being diagnosed. Not how many cases there were, but just what we were recognising at the time. And we found that about 5% of dementia diagnoses were being called dementia with Lewy bodies, which was much lower than we expected on the basis of neuropathology and all sorts of other evidence. And we also found that they had a difficult ride. They took twice as long to make a diagnosis as people with other sorts of dementia. It took over a year to get a diagnosis. And almost half were given the wrong diagnosis first. That's a huge amount. Half of people called something else first before dementia with Lewy bodies. Of course, around that time, we had a very famous person who unfortunately had the diagnosis really confirmed after death. Robin Williams, you know, great comic film actor, died by suicide in 2014. And his illness, which was clearly dementia with Lewy bodies, was not diagnosed during life, despite seeing several specialists, having inpatient care and all sorts of things. And his widow, Susan Schneider-Williams, who's been the most fantastic advocate for dementia with Lewy bodies and just sort of the most amazing person, writes, and it's an article I'd commend to everybody, "The Terrorist Inside My Husband's Brain," about how Robin had suffered with symptoms that we know are very similar to the kind of classic features of Lewy body dementia: depression, autonomic features, memory problems, fluctuation, thrashing around during sleep, REM sleep behavior disorder, struggle to remember a single line for a film, and the various other things around that time show that unfortunately it was very obvious to someone else, including people on the film set he was working, that there were difficulties. And he never got a proper diagnosis. He was unable to deliver lines, and yet, you know, people just put it down to something else. So that's sort of unfortunate, very. What can you do about diagnosis? That's all very well saying it's a problem. So we approached it by trying to develop some this is just sort of stuck. There we are. Some toolkits to try and improve diagnosis. So this was ways of reminding people in memory services that there was dementia with Lewy bodies, but more importantly, it was how you look for it. So it's all very well saying, oh, these people fluctuate, have hallucinations, have Parkinson's. And well, if you're a clinician, I'm really busy, I'm running a clinic, I've got, 15 minutes with somebody, what am I going to do? So we had single validated questions to ask about fluctuation, to ask about REM sleep behavior disorder, to look for Parkinsonism in people, and put them together in a toolkit that literally took, a minute or two to do. And then putting this into services, we were able to show you could increase rates of detection of dementia with Lewy bodies by using these toolkits. Whether it was the toolkits or just raising awareness, it didn't really matter. It was out there and people were starting to recognize it. And it was a 35% increase. Now, when we were doing this, or I was doing this sort of work, I was probably, well, not probably, I was kind of just obsessed with diagnosis. diagnose. And people kept on telling me, including grant-giving bodies who would turn down grants, saying, well, it's not all about diagnosis, what about management? What are you going to do with these people? And it took me, many years, I think, before I sort of realized, of course, they were right. Why would people ever diagnose something if you can't do anything about it? So then we switched to put more emphasis on the sort of management of it. Because although there are no disease-modifying treatments, there's actually, like with Alzheimer's disease, there's quite a lot you can do for people with dementia with Lewy bodies. And it's sort of different from what you do in Alzheimer's disease and frontotemporal dementia and vascular dementia. So we put together some management toolkits. John Paul Taylor, Ian McKeith, very involved with this, from a kind of combination of looking at the best evidence, so all the systematic reviews. We looked at others, we pulled that together. And there were a lot of areas where there was no sort of evidence. So what do you do when there's no evidence, but you've got patients and you want to do something about that, and you haven't got randomized controlled trials? Well, you do the next best thing, which is expert opinion. So we assembled 25 experts and put various suggestions to them as what would you do? Is this a good treatment? And we used the Delphi approach. And if more than 75% of people said, yes, this is absolutely what you should be doing, we then took that as evidence and put it in the toolkit. So it's a mixture of randomized controlled studies and expert evidence. And it's sort of divided into several domains. I'm not going to go through this in any detail, but we weren't just interested in the cognitive bit of the dementia with Lewy bodies. As important were the neuropsychiatric features, the hallucinations, the sleep disorder, the autonomic features, drops in blood pressure, urinary problems, and also the motor features of Parkinsonism, and about how we could help people. And it got sort of quite wide recognition. I was pleased that at one of the International Lewy Body conferences, some of our colleagues said it was the best thing at the conference. I think that probably tells you that there wasn't much else at the conference rather than this. But nonetheless, it was sort of nice, and it's been taken up more widely than I think we thought. So it's been taken up in the US by the Lewy Body Dementia Centres of Excellence, by the European DLB Consortium and others. So things that maybe you don't initially think might have impact sometimes do, and we're revising this to update it and get a new toolkit. And we've made it much more international. So we've got a Delphi panel, but it's much bigger and it's much more international, helping us with that particular work. Outcome, dementia with Lewy bodies. This is my colleague Annabelle Price in Cambridge, is unfortunately less good than in Alzheimer's disease, which is why we need better management. You can see here striking increase in mortality. I mean, it's just incredible, really, when you look at the figures. Survival after diagnosis in DLB is only about 3 to 4 years compared to 6 or 7 in Alzheimer's. And this is not just due to delay in diagnosis. If anything, that accounts for a relatively small part of it. But the main point of showing this is that, you can do these studies in different ways. I talked about sort of clinical studies, I talked about pathology, but this is a sort of e-record study. So it's using all the data from our Mental Health Trust in Cambridge over about a 20-year period all anonymised, lots of cases, hundreds, thousands of cases, thousands of AD cases, about 800 DLB cases, relatively unselected in the sense it's everybody's data. I mean, obviously there's selection to get to the hospital, but it's not like you're getting people who have to consent to a research study, all the governance, etc., all the approvals in place, and able to show in sort of naturalistic just how significant this effect is. Again, I mean, e-records aren't the answer to everything, but they can, they can help, and it's another source of data that can be very useful. To do a clinical trial in DLB, we're doing one at the moment on memantine, the cobalt study. It takes a lot of money, it takes years, it takes a huge amount of effort. Now, you can't ever short-circuit a randomized controlled trial. That's the gold standard, and we need those. But you can look and see what other sources of data there are. So we looked in this same data set, the e-record data set from the Cambridge Peterborough Mental Health Trust, and looked if there was anything that seemed to influence mortality, because mortality was so poor in DLB. Was there anything that gave a signal that it might actually improve things? And there wasn't a lot, but actually treatment with anti-dementia drugs was one thing that significantly did increase mortality. And it's always difficult in a study like this to have a control arm, because the people who get the medication are not matched to the people who don't get the medication. There's sort of choices along the way. But nonetheless, you can try and match for comorbidities and things as best you can, and certainly show that if anything, treatment has a beneficial effect, certainly not a harmful effect. And I think things like this were helpful. This e-record study was relatively easy to do because the data is just sitting there. And at the time, and I think, even since then, really, people have always been concerned about long-term therapy in people with dementia with Lewy bodies who have kind of cardiac and other problems. And this at least shows it's safe, but actually beneficial, keeping people on medication for many years. In terms of sort of other things moving forward, well, inflammation we'll hear about quite a lot at this conference, and we've been interested in this in dementia with Lewy bodies. Again, a different modality, just to illustrate another way of looking at things. This is PET imaging using what's quite an old ligand now, but quite a robust one, PK1195, which is looking for microglial activation in people with, in this case, dementia with Lewy bodies. And it's been shown across a number of neurodegenerative conditions: Alzheimer's disease, frontotemporal dementia, Parkinson's disease, progressive supranuclear palsy, that inflammation seems to be an important factor and may be a driver of disease. And we looked at it in dementia with Lewy bodies and found there was indeed an increase in binding of this ligand, so increase in microglial activity as we interpreted it. But it was limited to those with early DLB. In those with more severe, but these were all mild to moderate cases. But once they got beyond the mild stages, it looked like the inflammatory response had kind of reduced, which is interesting. And this is something we're following forward within the Dementias platform, which again, has been a fantastic source of structure and support and networking, like this meeting is. And within the current Dementias platform, within experimental medicine bit, we have a neuroimmunology work package where we're looking at this. And my colleague Mara Malpetti, who has been looking at this in a group of people across diagnosis in something we've called the Nimrod study that we've been running on Cambridge for some years, which has been looking at PK imaging and blood biomarkers of inflammation in Alzheimer's, Lewy body, frontotemporal, progressive supranuclear palsy. And I won't go into the details, but we've shown, and she's got a paper in press about this, Rob Durkin, that not only can you develop, or can you demonstrate an increase in inflammatory markers across different diagnostic groups with some similarities and some differences, but the effect of this on outcome, and I mentioned mortality before, is significant, such that people that have sort of certain increase in these markers have significantly increased mortality compared to those who don't. So these are things, because these are things that might be amenable to intervention. So I think they're particularly interesting. And we're carrying on looking at this through the Imprint study, which is part of DPUK, and Peter Swan, Leon Hararo, I think, are in the audience, or certainly coming to this meeting, who've been pioneering that. And I'm kind of reflecting a bit what Atticus and Simon said. I mean, early career researchers are the future for developing novel methods. There's looking at very new methods to look at particular cell subtypes rather than what we've previously done, which is look at cytokines and general measures to see if there are particular profiles that differ between people with Alzheimer's disease, Lewy body dementia, and controls, and focusing much more on sort of early disease. And Alina's got a blog at the moment on the Dementias platform. Please have a look at it, about mild cognitive impairment due to Lewy body dementia, which is Lewy body disease, I should say, which is relatively recently described as a form of prodromal DLB, so sort of moving into the early space. So there we are, lessons learned from Lewy. Well, history and terminology matter. I think we need to pay attention to them. I think one thing I've learned is translating research into clinical practice takes a long time, but it can be done, and it is very important. I mean, that's why we do research, is to make a difference to people. I think I've learned, and I'd encourage you to think broadly about which research methods best answer the question. We all maybe just stick with what we know and think that's the answer, but sometimes the question we want to ask is better answered by other types of data or other methodology. For example, it might not be a clinical study, it might be pathological, might need imaging, genetic biomarker, maybe you need a trial, maybe you can look at existing data, dementia portal, etc., to answer the question. So think broadly. I think collaboration is fun, rewarding, and it moves research forwards. So I would encourage people definitely to collaborate. You don't need this because you're all here in person, but some people are reluctant, I think, to come to meetings, and I think we need to extol the benefits of in-person meetings. That is absolutely tremendous. Another thing is don't throw samples or data away. There's the DPUK portal for putting your data on. I actually got an email this morning from somebody who had been working on a data set we had. In fact, she'd done her PhD thesis on it 20 years ago. And she said, I've just thought of another question. We can use this data for a student she's supervising. And it's absolutely perfect for that particular question. So, studies and data, old samples, biosamples, they can be incredibly valuable with new methods. And support yourselves. We need to support you. And that's also never too early to start. I think when I was an ECR, I kind of thought I was an ECR for a very long time. But actually, as soon as you're an ECR and you've got a couple of years' experience, you can help other people, you can help students, you can support others. So in a sense, I'm sure you are, but it's kind of never too early to start. It's very rewarding, and you, as Atticus clearly said, are the future. So enjoy the meeting. It's wonderful to see you all here, and I think we'll have a great couple of days. Thank you very much.
But the advice we give to all ECRs is to follow the Involve guidelines for kind of funding. And but one of the things I'd say is reach out to the Alzheimer's Society, because we have our research network and we have a kind of partnerships opportunity where people who researchers who aren't funded by us can kind of can use our research network, and we have a number of people living with dementia within that network as well. So kind of if you with that costing, and we can help with that costing guidance as well. And our research network for our funded researchers, they do it all kind of a lot of the time for free as well. So we can explore that as an option. But I would recommend going to the Involve guidelines, looking at those at the first instance, and then kind of getting in contact with us as to what exactly you want. And the involvement team will be able to help support that.
I'd also add that exactly that around the involved guidelines. Unfortunately, it can be expensive, but there are kind of grants or opportunities available to support with some of those costs. And if you're applying to kind of us as funders, we'll consider those. And also reach out to kind of our network centre, because they'll have, even if they don't have necessarily a PPI group, they might know of people that have groups set up with kind of those people that you're looking for in them. So it's kind of getting the word out to see who knows what group and where, and how you can use sort of existing groups like the Research Network, but also others.
Great, thank you. Anyone else with a question? Yes.
John's touched on the people with Lewy body dementia have a toolkit, but all of the research seems to be preventative or curative. What's been done for those, 1.2 million at the moment, they've already got a diagnosis?
Well, we fund research into care. So, for example, we recently funded our doctoral training centres, and one of the centres is on integrated care, and that's 3 million. We recently launched our health inequalities call, which is care focused, and that's a 2 million call, and kind of we'll be announcing that as well. So we fund research into care. We currently have research into care. It's not the largest part of our portfolio, but we have a kind of a percentage of that within it. So we are really committed to kind of improving the lives of people currently living with dementia, and we do fund that research as well as offer the kind of support services as well. And what's really good about kind of the Alzheimer's Society is we have those support services, and we also have the research that we're funding into care, and we're trying to see how we can marry the two together, how the research that's happening is influencing the support services that we're seeing and the dementia advisors that are going out and kind of in the front line. So yeah, we do fund that research for those people living with dementia currently.
I was on a recent project for the Sage House project in Tangmere, and the qualitative analysis that we've done is now going through replication to suggest that people with a dementia diagnosis not only feel abandoned, but they feel that there's no access to care within any type of dementia research. And again, things like the Sage House project in Tangmere shown to reduce the government spending by 1.6 billion, but unfortunately, when we put forward models like this, treatment as usual models, tend to kind of say, well, we're just doing diagnosis at the moment, we're not offering people support. So again, I think that sometimes the diversification of our research should really be to target those people who actually have this diagnosis, are living with this condition, that are forgotten, that are left to disintegrate to the point of needing to be re-engaged within therapeutic interventions.
Could I just add? I mean, I totally agree. I totally agree with you, and I think one thing that's happened with a lot of industry but also non-industry funding is it's very much gone in the front end, and I've often bemoaned the lack of let's take clinical trials in people with dementia. We have so many key questions we need to answer, and that was why I put a lot of energy into the Diamond Lewy toolkit, because it's people with established dementia going all the way through to severe. So I think there is a sort of imbalance. I don't know if Prof Malhotra's here, but he's certainly at the conference because he's speaking tomorrow, and he's going to talk about a platform that he's developing, which is for people with dementia, and I think that's so important that we do. Whatever happens at the early stage, whatever fantastic things happen in terms of the MABs or what's coming after them, there are going to be a lot of people with different types of dementia, maybe even some of them, unfortunately, having them for slightly longer, and we absolutely have to address that.
Thank you all for your great presentations. My question is probably for John O'Brien. So you mentioned that the translation of research into adaptation for clinical guidelines can take up to 20 years. So I was wondering, for early career researchers, if you have any advice on what we can do to potentially expedite that process. Would you suggest, for example, to be involved in clinical applications early on, or to speak more to people in the clinical field? Are there any tips that you would suggest, for this timeline to be expedited?
No, thanks for that. I mean, I think again, you look back on mistakes you've made yourself and what you wished you'd done. And I think what I'd maybe wished, although, you know, I've been involved with NICE for many years, but somehow when I was setting up my studies, I didn't have them as my endpoint, and I should have done, because they operate in certain ways. I mean, I know it's very UK specific, England and Wales, I should say, specific, but nonetheless, if you want your research to ultimately change clinical practice, you've got to know what their thresholds are and what they will need. And there's no point designing a study that we did in good faith and thought it was a great study. And then NICE said, well, actually, that's not really what we're looking for. We're looking for something else. I don't think they changed their mind. I think that was always the case. So, you know, with regulators, it's early discussion. You can't really discuss these things with NICE, but you can see what they need in terms of what they would want as sufficient, both sufficient evidence and the right sort of evidence to change things. And then they'll take account of it. But in the last dementia guideline, for example, as just one example, they didn't like any of the studies on amyloid imaging that had been done because they didn't meet the right criteria, because they've been sort of autopsy validation type studies, and they wanted something that was much more clinically focused in terms of showing how people with uncertainty would benefit from getting an amyloid scan. And now at the time, those studies weren't available and done. They have sort of been done since then, but that was one example where, though there's quite a lot of evidence for amyloid imaging, we were all convinced by it. It didn't meet their threshold, despite all the effort that had gone into that.
My question's to Emma. As someone who works in such a rich data environment, what would you recommend for ECRs who are collecting data and would like their data to be as useful as possible to other people? I think we start off quite small and just think about what we need and what we can do, but what can we do looking forward? And a second thing, if that's okay, as a female leader in the field, what would your career advice be?
Just the first question first. So I think a good start is to look at the studies. You can even look on the data portal website at studies that have already been collected and how they've been collected. And that gives a good idea, particularly when getting consent for onward sharing at the end. It's important to collect that at the beginning. It saves you a lot of paperwork further down the line. So there's lots of advice on the online to be able to set up those studies. But I think if you're very early stages, reaching out to people like at DPUK Data Portal and saying, I'm at the beginning, do you have any advice? And we can reach the data analysts who would, you know, use the data, and I'm sure they've got recommendations to make that data set a lot easier. And sorry, I missed the second question.
Oh, I was just saying, as a female leader in the field, what's your career advice, given that we've heard from two men about their career paths? What do you recommend?
Surround yourself with really clever people. That's it. I mean, my background personally was I started off in genomics data and worked in a wet lab, and I was far better at organising things, processes, and moving those things forward. But I was absolutely convinced that I should be in research, but it wasn't for me. It really wasn't for me. I'm really good at making things more efficient, and luckily, I've hired really clever people as well. And I surround people like neuroimaging experts and genomics experts to be able to do that. Pick your network wisely. I think that's, you know, reach out as much as you can and build that network, because your network determines what you do, and the most random ideas will come in the most random places and push that forward. And I think Simon Young, do what you enjoy. And I love making things efficient, and I really enjoy being having a team and working with a team to deliver something that has meaning and purpose. That's probably a bit for me.
Thank you, Emma. We actually did have a female speaker about their career trajectory, and unfortunately she couldn't make it. So, so thank you, Emma. There we go. Emma stepped in there. Any other questions?
Sorry to hold everyone from their break. I'm Seb Walsh. I'm a public health researcher at Cambridge. I work with Carol Brain, who John will know well. I was just reflecting that we have heard about the importance of bringing together data from clinical samples, Neuropath, and of course we also have that kind of community-based, population-based evidence. We haven't had prevalence estimates since CFAS, really nationally representative ones. They're now 15 years old. I'm not aware that there's any real plans to do another national prevalence study. So I just wonder if we feel that's a gap, how important and how useful would it be for some of the other research endeavours we're talking about to actually have that solid population denominator from representative samples? And if we agree that's a gap, how might we go about filling it?
Thank you. Anyone on the panel want to?
Well, I think we learnt so much from CFAS 1 and 2, and we've learnt a lot from other similar epidemiological studies, thinking of ones in the US and Sweden that have sort of gone back and looked at prevalence and incidence of dementia after 20 or 30 years and shown very striking findings. As you know that actually things have decreased, more dementia around because they're more older people, but actually better education, vascular care, whatever, has led to reduction. So I think we learn an awful lot about what might be going on in terms of risk factors for dementia, and I think we learn a lot about cases. So in an ideal world, yes, absolutely, we would want these to be repeated every 20 or 30 years, 20 years probably, so that we get a good feeling of what's going on. The downside is expense, I guess, and competing with other studies and the additional benefits. So, I mean, I'm not the person that makes these decisions, but I can just see that budgets are tough, so what do people put resource into? But the value of them is sort of unquestioned. It's just whether it's enough to take things from something else. That's all I can say, really.
Okay, so I just want to thank the panel and thank all the speakers this morning and thank all of you. Excellent attendance, and thank you very much. I really hope you enjoy the rest of the conference, and please do reach out to any of the speakers today over the next 2 days. Also me, if you've got any further questions or just want to say hello, but really just enjoy the conference. Thank you very much.